First complex you need to know about in A2 chemistry is Tollens reagent. Tollens reagent is a test for aldehydes. Tollens reagent is a complex with silver attached to two ammonia ligands and has a positive charge. The silver has a 1+ oxidation state. There are two coordinate bonds between the silver and the two ammonia ligands, as you can see here, and it's 180 degrees. So as I said, it's a test for an aldehyde, and when the Tollens reagent reacts with the aldehyde, the following reaction takes place. So the silver in the Tollens reagent is reduced because it gains an electron, and it turns into silver solid and releases two ammonia molecules per complex. At the same time, the aldehyde is oxidized to form a carboxylic acid, and you can see the equation here. Aldehyde reacting with water, which is in aqueous, from the aqueous solution, forming a carboxylic acid, 2 H+ ions and 2 electrons, so it's lost electrons, so it's been oxidized. So the observation of the reaction is that there's a silver mirror on the test tube, which is due to the silver solid precipitate that's been formed, as you can see in the equation here. And just a little summary of how Tollens is made, which you don't need to be aware of, but sodium hydroxide reacts with silver nitrate, which forms the brown murky precipitate of silver hydroxide. Ammonia is added to this precipitate. Only a small amount is required, but it's concentrated, which forms the Tollens, and the ammonia reacts with the silver, forming the complex. So iron is another metal which forms a complicated complex called hemoglobin, which is used to carry oxygen around the body. Oxygen forms a coordinate bond with the iron, and the oxygen is then carried around the body to the cells. There is a coordinate bond that forms between the oxygen and the iron, as you can see in this very simple diagram here. The hemoglobin complex is a rather complicated one, with iron in the centre, and then it's connected by something called a porphyrin ring, and then a myoglobin protein on the top. So it actually forms a coordination number of 6 when you take into account the oxygen. Carbon dioxide replaces the oxygen, which the cells release, but then once the carbon dioxide has been lost by the iron complex, oxygen can then form a coordinate bond with the iron again, and this process is reversible between carbon dioxide and oxygen, which is why this process can continue without any problems. Carbon monoxide, on the other hand, this is a non-reversible process. The bond between the carbon monoxide molecule and the iron, the coordinate bond, is extremely strong. It's 230 times stronger than between oxygen and iron, and so this coordinate bond is not broken, and that is why carbon monoxide is a highly poisonous chemical. Cisplatin is another complex you need to be aware of. It's a square planar complex with platinum, 2 ammonia ligands and 2 chloride ligands. The platinum has a +2 oxidation state. It's used in the treatment for some cancers. Its action is the cisplatin complex reacts with water, which is found in the body, and 1 chloride ligand is replaced in a ligand exchange reaction. The platinum then reacts with one of the guanine base pairs, and water is lost. Then the complex reacts with another water molecule, and the complex B, I call it B, this complex reacts with a second guanine base pair on a different strand of DNA. And what this does is it interferes with the cell division of the cancer cells.